Okay guys, today I continue with the series of the antibiotics and today I'm talking about the quinolones and the fluoroquinolones. Starting from a very basic question, what's the difference between the quinolones and the fluoroquinolones? Quinolones were the first drugs which are found in this series. The example is naldixic acid. Okay, naldixic acid was found to be effective against some gram-negative bacteria, especially those causing the urinary tract infection. And because this drug is concentrates well in the urine, it was used for UTIs. But the problem with this drug it has a very limited or a small spectrum against the bacteria. It was killed only few of the gram-negative bacteria. And secondly, this drug is not well distributed into our tissue or our body fluids. So this drug was not so effective as an antibiotic, and it was an important and a small group of the drugs. But then something good happened in the chemistry labs when they added fluorine into the quinolones, and this we can say fluorinated quinolones. Fluoroquinolones, it means fluorinated quinolones. These fluoroquinolones have a very extended spectrum of action, and also they are well distributed into our tissues. These fluoroquinolones currently are very popular and commonly used drugs. Why these drugs are very commonly used? There are many reasons. For example, these drugs are usually orally active. They can be given orally, and also they are well absorbed. These drugs are administered orally. Also, after oral administration, they are very well absorbed from the GIT and goes into the circulation. Secondly, the fluoroquinolones are very well distributed. The fluoroquinolones are well distributed into our tissues and not only killing the microorganisms or the susceptible pathogens in the extracellular fluid, but they also kill intracellular organisms. These drugs penetrate into the neutrophils and the macrophages to kill the intracellular microorganisms. And they are broad spectrum antibiotics. So it means what? They can kill many microbes, gram-positive, gram-negative bacteria, also some intracellular organisms due to penetration into the neutrophils and the macrophages, and also some of them can kill some anaerobic bacteria. And finally, why these drugs are so commonly used and popular? They are safe drugs. They have side effects, but usually not severe. And if they have severe side effects, the side effects will be what? Will be very, very rare, not common side effects. Okay, so they are regarded as safe antibiotics. But what's the mechanism of action of these drugs? How they work? These drugs are usually regarded as what? As bactericidal. What do we mean by bactericidal? It means they can kill the bacteria. Fluoroquinolones are killing the bacteria, and they are even effective in low concentrations. Their killing effect is concentration dependent. It means by increasing concentration, more of the bacteria will be killed and faster. But even though they are effective in very low concentration to kill the bacteria. So what's the mechanism of action of the fluoroquinolones? Quinolones on the molecular target, they inhibit the topoisomerase or topoisomerase enzymes, group of enzymes. But what's the topoisomerase? What's the role in the biology of the bacteria? Let's go into this. You know that bacteria has a chromosome, of course, right? And the bacteria chromosome is a double-stranded DNA. It's a circular double-stranded DNA. Okay? If we enlarge part of this DNA or a gene in this DNA to see what's happened there, I bring it here to here. This is the double-stranded circular of the DNA. And bacteria, for the growth of the bacteria, needs what? Needs proteins. And for making proteins, the bacteria needs what? Messenger RNA. How produce messenger RNA? From the DNA transcription. Part of this DNA is transcribed into what? Into messenger RNA, and this messenger RNA, with the help of the ribosome, will make what? Proteins. And also, the bacteria needs to replicate. For the replication purposes and division into two daughter cells, the bacteria first of all needs to replicate this DNA. So for the replication purposes or the transcription purposes, this double-stranded should be separated and making new copies, right? So it will be right. This double-stranded first of all should be separated. Of course, there are nitrogenous bases. If bacteria is going to replicate, it needs to make another copy of this DNA. It means one copy of this strand and one copy of the other strand. And for the transcription, for making messenger RNA, it will make a copy from one strand, either this one or the other one. So what will happen? After separating this area, this double strand, because this is a circular chromosome, circular DNA, and there is no free in this. Bacterial chromosome has no free in this because it is circular, right? After opening or separating this double strand, this area will be loose, and this area will be more tight, right? The double strand of the DNA are wrapped around each other. They are twisted around each other. By opening this part, the other parts will be what? Will be more twisted, will be more wrapped around each other. The number of the twists will be increased. So when the bacteria needs to replicate or transcription of this double-stranded DNA, after separating this area, it cannot proceed more, it cannot move forward because this area will be very, very tight, and this area also will be very tight due to increasing number of the twists. And this is the mechanical issue for the bacteria. How it will solve it? Then some other enzymes will come to here. Okay, this enzyme will come to here. And this has two domains. One cutting domain, breaking domain, and another domain with different functions. Okay, and also another enzyme here. Okay, this enzyme will solve the mechanical problem with the twisting. After opening, separating this area will be over twisted, okay? And this area will be loose or opened, and this area is over twisted. And so the replication of the transcription cannot continue due to mechanical obstruction, okay? Let me enlarge it here so you can see how this enzyme works. this is our enzyme. What's the function of this enzyme? This enzyme has a breaking or cutting or a nuclease domain. This nuclease domain will make a break here in one of the strands. After cutting this strand, what will happen? It will untwist. It will pass through the other strand one, two, or three times around the other, and the over twisted DNA will be what? Under twisted due to making one break in it, and this have a free end. Also here there's another free end, and this free end will pass through the other strand of the DNA. And after it was under twisted, what will happen? The other domain will come to action. This domain is let's say rejoining the breaked strand of the DNA, or we can say resealing, rejoining, ligating. This is a ligase domain that can rejoin the breaked DNA after it intertwists or passes through the other strand. So this rejoining is occur at the same point when there is breakage, so there will not be any mutation or change in the primary sequence of the DNA. So what's the name of this enzyme? This enzyme is called topoisomerase, and especially it's topoisomerase 2. Also it's called, in the bacteria it's called what? DNA gyrase. So again, what's the function of the DNA gyrase or topoisomerase 2? From the bacteria there is circular double-stranded DNA, and for the replication purposes or the transcription purposes, this double-stranded should be separated at any point, okay? And after separating this area, or at the point where the DNA strands are separated, it will be lost. The other part of the DNA double-stranded will be over-twisted, or we can say supercoiled, or sometimes it's called positive supercoiled. Positive supercoiled, it means it's over-twisted, and more it will be wrapped around each other. Now the DNA gyrase or the topoisomerase will come to here and make a break in one of the strands, and after making this break, there will be free ends of the DNA. At this free ends, it's going to pass through the other strand, and it will be what? Under twisted. Under twisted is called what? Negative supercoiling. So you can say the function of the DNA gyrase or topoisomerase 2 is to introduce or producing negative supercoiling in double-stranded DNA of the bacteria. And after under twisting of the double strand of the DNA, the other domain will come to action, and this domain is resealing or rejoining the breaked DNA. And in this way, the replication or the transcription of the DNA can be continued to make messenger RNA. But what's the action of our drugs, the fluoroquinolones? Fluoroquinolones will come to here, and after diffusion into the bacteria by passive diffusion, they reach the topoisomerase 2, and by approaching the topoisomerase 2, they will inhibit or bind to this domain. Okay? Fluoroquinolones will bind to this domain. What was the function of this domain? This was the ligase domain, or the resealing domain, joining domain of the DNA strands. And by binding to it, the topoisomerase or the DNA gyrase is continued to make rings, cut the DNA strand, pass through away under twisting, but at this time it cannot reseal it. Normally it functions to what? Making a cut under twisting and rejoining, making another cut under twisting and rejoining, and so on. But the fluoroquinolones will bind and block the ligase domain, so the topoisomerase still continue to what? To breaking the DNA strides. The nuclease domain will not be inhibited, and the topoisomerase is continue to break, but without resealing, all the DNA will be what? Will be fragmented. So fluoroquinolones leads to what? Fragmentation of the DNA. And with the fragmentation of the DNA, of course, you know there will be no replication to daughter cells or no transcription to messenger RNA, and eventually the bacteria will die without growth and without replication. The DNA gyrase is an important target, especially in gram-negative bacteria. Okay, it's more important in gram-negative bacteria. The nalidixic acid and even the second generation of the fluoroquinol ciprofloxacin are more effective against the gram-negative bacteria, but the newer agents and new generations of the fluoroquinolones are more effective against gram-negative bacteria and also gram-positive bacteria. Then they found in the gram-positive bacteria it appears to be another target for test drugs, and it was called topoisomerase type 4. In the gram-positive bacteria, there's another enzyme which is called topoisomerase type 4. So what's the function of the topoisomerase type 4? After replication of the DNA, let's suppose this is a double strand of the DNA, okay? This is part of the double strand of the DNA, and this is another double strand. Okay. Here we have another one. Okay. What are these? After the replication is completed, many of the double strands of the chromosome will be interlocked within each other. And another enzyme will come to here, which is called what? Topoisomerase, but which type? Type 4. Okay, it has another a nuclease or cutting arm and also a rejoining arm. This topoisomerase 4 breaks the DNA strands at 2 points. Both of the strands will be cut. The topoisomerase 2 making a cut only in one strand of the DNA, but the topoisomerase 4 making cutting in both strands of the DNA. So, for example, by breaking here, okay, making two cuts in the blue DNA double-stranded, it will takes it out from the black one and then rejoin it. So in this way, it removes the entangled double-stranded of the DNA. The double-stranded are after replication is usually entangled within each other, but by making it breaking and taking it out, and after that rejoining them, remove the entangled double-stranded of the DNA, or medically this is called what? Catenated DNA. Catenated means what? Means all the double-stranded of the DNA are entangled, interlocked within each other, but the topoisomerase 4 leads to decatenation of the DNA, removing the interlocked double strands of the DNA. Or let me make it in another way. If there's one double strand here, okay, and another one here, these two circular chromosomes of the DNA are interlocked within each other, it will make a break from one of them, okay, removing the other one from that break, and then it will be like what? It will be two separate chromosomes. So what's the function of the topoisomerase 4? It is used for decatenation of the DNA, which removes the interlocked double-stranded DNAs from each other. And what's the function of the topoisomerase 2? It untwists or under-twists the over-twisted DNA, okay? Or we can say it introduces negative supercoiling into a positively supercoiled DNA. This is the basic mechanism of action and the molecular mechanism of action of what? Of fluoroquinolones. We said that DNA Gyrase is more important in the gram-negative bacteria, but the topoisomerase 4 is more important in what? In gram-positive bacteria. That's why the other agents such as nalidixic acid and ciprofloxacin have limited activity against the gram-negative, the gram-positive bacteria, but the newer agents are more effective against gram-negative and also gram-positive bacteria by blocking or inhibiting the action of the topoisomerase 4 from its ligating domain. They only inhibit to this domain. This is about the mechanism of action of the fluoroquinolones. Now let's talk about the details of their clinical uses. Okay. Okay. Generally, the fluoroquinolones and the quinolones are divided into 4 generations. The first one, we have first generation. Okay. We have second generation. We have third. And of course, we have fourth generation. Okay. The first generation are basically not fluoroquinolones, they are quinolones. Okay, they should be called what? Quinolones. The example of the first generation is the nalidixic acid. Nalidixic acid is primarily used for urinary tract infection due to its activity against some gram-negative bacteria that causing the UTI, such as E. coli. So you can say it is moderately, okay, not so active, moderately active against gram-negative rods, such as E. coli or Proteus, and its use is only limited to the UTI, and currently it's not used commonly due to more effective choices and also safer drugs. Nalidixic acid was only active against some gram-negative bacteria. Example of the second generation, such as what? Ciprofloxacin. Of course, others such as levofloxacin, moxifloxacin, and many others. Okay, these drugs, which are fluorinated fluoroquinolones, they should be called what? Fluoroquinolones. The first generation is not a fluoroquinolone, it's a quinolone drug. But the second generation, third generation, and the fourth generation are the fluoroquinolones. Fluoroquinolones such as ciprofloxacin, which is regarded as the prototype of the group, as effective against more gram-negative rods. Okay, ciprofloxacin is active against more gram-negative rods, and it also have activity against some gram-positive cocci. Okay, but within this gram-positive cocci, one of the most important pathogens from the gram-positive ones is not covered, and it's called what? Streptococcus pneumoniae. Pneumococcus will not be covered by the ciprofloxacin, and that's why ciprofloxacin should not be used for pneumonia or community-acquired pneumonia when there's the pneumococcus is a common pathogen. So ciprofloxacin is not regarded as a respiratory quinolone. Ciprofloxacin is only active against some gram-negative rods and also some gram-positive cocci, but not including the pneumococcus. The gram-negative activity of the ciprofloxacin is far more than the gram-negative activity of the nalidixic acid. Then we come into the third generation. Example of the third generation like what? Levofloxacin. Levofloxacin retained the ciprofloxacin's activity against the gram-negative rods. It's again active against many gram-negative rods. Okay, but it's more effective against gram-positive. Many gram-positive cocci are covered by what? By the levofloxacin, and including the pneumococcus. Okay, levofloxacin is not only effective against the gram-negative rods, but also many gram-positive cocci, including the pneumococcus. Ciprofloxacin and also levofloxacin are also active against some atypical organisms. Atypical organisms like what? Like mycoplasma, chlamydia, also Legionella, Mycobacterium, Brucella. Okay, again, the third generation levofloxacin also effective against this atypical microorganisms. Atypical microorganisms such as Mycoplasma, Mycobacterium, Legionella, these are regarded as atypical and intracellular microorganisms. Ciprofloxacin is not only effective against them in vitro, but in our body also effective due to its penetration into the neutrophils and also the macrophages. These atypical microorganisms are regarded as intracellular organisms, right? And the ciprofloxacin, levofloxacin, or other fluoroquinolones have the ability to penetrate into our cells, so they can effectively kill these drugs. But why they are called atypical microorganisms? Chlamydia, Legionella, Brucella. Why they are regarded as atypical ones? Because they cannot be stained well by the gram stain. Okay, and these are the spectrum of the levofloxacin gram-negative rods, more gram-positive cocci when compared to the ciprofloxacin, and including pneumococcus, also atypical microorganisms. And about the fourth generation, What's the example of the fourth generation? Fourth generation, like, most importantly, moxifloxacin. Okay, moxifloxacin is retaining the gram-negative activity of what? Of levofloxacin and ciprofloxacin. Moxifloxacin is also active against many gram-negative rods and also even more effective against gram-positive cocci and pneumococcus. Okay, and also atypical microorganisms, or less importantly than atypical, also it has activity against some gram-positive rods. Okay, and finally it has activity against some anaerobies. So moxifloxacin is even more effective than the levofloxacin for what? For pneumococcus. It also covers the gram-negative rods, atypical microorganisms, such as Legionella, Mycobacteria, and also some anaerobies like Bacteroides fragilis. That's why moxifloxacin is also used in mixed infections when there is mixed infection of the gram-positive, gram-negative, and also anaerobics. Moxifloxacin and the levofloxacin are regarded as respiratory fluoroquinolones because they are active against pneumococcus, which is the main cause, the most common cause of community-acquired pneumonia, and any drug which is to be used for the pneumonia should cover the pneumococcus and also some of the other microorganisms. But because the ciprofloxacin will not cover the pneumococcus, it's not regarded as a respiratory fluoroquinolone and not used for what? Pneumonia. This is a basic idea about the differences in the generation of the fluoroquinolones and their spectrum of action.